Good evening, good afternoon, everyone, wherever you might be. I am Rob Burgess, Chief Business Officer for Sino-Biological, and it is my pleasure to welcome you to the next installment in Sino's Lock and Key webinar series on cutting-edge technologies which often employ the use of Sino-Biological products and services. So just one main housekeeping issue before I introduce our speaker for today. If you don't mind, if you have questions, I encourage you to enter those questions into the chat box, and we'll hold all questions until the end of the seminar. And then I will run through the chat box and get to as many questions as I possibly can. So again, just type your questions into the chat box and we'll get to them at the end of the seminar. And so with that, it is my pleasure and honor to introduce to you today our speaker, Dr. Samuel Tia. Dr. Tia is co-founder and chief scientific officer of Correlia Biosystems. He works there with our scientific teams to expand Correlia's immunoassay test menus and deliver customer solutions for proteomics-based applications. Dr. Tia received his doctorate from UC Berkeley, where he worked in the HER lab and actually developed a patent and licensed that patent out from Berkeley. In addition to that, he obtained his Bachelor of Science in Mechanical Engineering from the University of Florida, and he has very extensive experience in industry with a particular focus on drug delivery and medical grade manufacturing and packaging. The title of Dr. Tia's seminar today is The Ultra-Efficient Pharmacokinetic Measurements in an Automated Microscale Format. So Dr. Tia, it's a pleasure to have you today. I'm going to now mute and turn the screen over to you for your presentation.
Okay, great. Thanks, Rob. Appreciate it. It's a pleasure to be here today. Let's see. Just share my screen. All right, are you seeing my title slide now? Okay, perfect. All right, it's a great pleasure to be here this morning to be able to share with you some of the work that Correlia has been doing. This year, 2022, is going to be a big year for Correlia. This year, we're introducing a new platform for protein measurements, and we're really excited to show off some of the great data that is coming off of this platform. At the SLAS conference in Boston in February, we're going to be really doing the introduction of this product, but we can give you a sneak peek at some of the great data that we've been generating with partners and collaborators. This is one of the first time that we've shown some of these results in a public forum. And so I'm happy to be able to share, the content with you with a particular focus on a couple of case studies, specifically how Correlia's technology can be used and a helpful tool in pharmacokinetic studies, especially with small animal models. Here's an outline of my talk for today. We're going to talk about Correlia's Pixie platform. That's the immunoassay platform that we're introducing this year, which can really overcome a lot of the limitations that are associated with traditional immunoassays. The Pixie platform is built around a nanoporous substrate, which accelerates the rate of protein capture and measurement. The platform allows for increases in efficiency, plus measurements with broad dynamic range and high precision. Importantly, the Pixie platform also enables microscale sample measurement capabilities, which really opens up types of protein measurements that could have been previously inaccessible if relying on traditional immunoassay techniques, as well as the possibility of collecting higher quality, more meaningful data from your samples. There's no way we can cover all of the possible application areas today, so we're going to focus on a couple of case studies that the Correlia team has worked on with partners and collaborators. Specifically, we'll talk about how the Pixie kits can measure generic forms of IgG from small animal models, as well as more specific measurements for particular IgG idiotypes based on the associated ligand or the IgG subclass. And we will show some comparison data showing head-to-head results against traditional ELISA method and the MSD immunoassay platform. I would like to start at a higher level, though, and just talk about the importance of protein data in having a complete understanding, holistic, or should we say holomic view of biology. So over the last generation, everyone knows how nucleic acid measurements, gene sequencing has really revolutionized our understanding of human health. That's a rightful acknowledgement, and a lot of that progress came with revolutions in nucleic acid measurement technology, right? You can only study what you can measure. Now, at the same time, there's this other half, this complementary half of molecular biology on the protein side that has not advanced as quickly. And so Correlia is trying to do for protein measurements what Illumina was able to do for nucleic acid measurements, to advance the state of the art by providing better measurement tools. I think for us to really understand some of the current limitations of protein science, I think it's important that we discuss the workhorse behind a lot of quantitative protein measurements right now, which is the ELISA method. I think for a scientific audience, most of you are probably already familiar with this technique, but I will just go over some of the basics and some of the limitations of the method. So it's a workhorse, well-established quantitative protein measurement method that is built around a plastic microscale plate. So it's a pretty low-tech method, right? The method has remained relatively unchanged over the last 30 years or so. So you have these plastic plates, you have to fill them with sample. There's all these manual processing steps that are laborious, and you need trained people to be able to execute this. I mean, there are things that you can do to speed things up slightly. There's, plate manipulation systems that will handle your plates robotically, robotic plate washers, etc. But the basic limitations are with, long assay steps, long incubation times, and multiple manual processes that are prone to error. Next, we'll go over how Correlia's technology is able to address each of these individual problems, but I'd like to start with an overview of some of the basic principles that govern the majority of immunoassays today. So in order to do that, let's start by zooming in on an individual well, a unit cell of this 96 well plate. I'm going to use a 96 well plate as an example, but really this could be any immunoassay substrate. It could be the surface of a bead. It could be a nitrocellulose sheet. I think one of the basic limitations of traditional immunoassays is that they depend on the random motion of a target molecule in a liquid to collide with its binding partner. So in physics, this is known as Brownian motion. It's the three-dimensional random buzzing of a particle in space. And ELISAs depend on this random motion to encourage target binding between the antigen and the antibody. So in this particular cartoon, antigens are shown as green triangles coated on the bottom of the plate, and the antibodies are shown as the Y shapes within the well. And in order to facilitate an ELISA, long incubation steps are required, where you're basically, maybe you're rocking or shaking the plate in order to encourage the diffusive interaction between some of the antibodies and the capture molecules. Now, this will work to some degree, but it's never going to be 100% efficient. Also, it is an equilibrium process because at the same time when antibodies are binding to the bottom of the plate, antibodies are also being disassociated from the bottom of the plate. So you have to wait for a significant amount of time for it to reach an equilibrium state. There are things that you can do that will slightly accelerate the rate or make it maybe a little more homogeneous by maybe you rock the plate, right? Those are small changes that can help a little bit, but in the end, you're always going to need these long incubation steps, hours required. I think anyone who's done an ELISA knows how painful that process can be, a lot of waiting involved, and large sample volumes are involved. The characteristic length scale, or the interaction distance between the antibody and the antigen, can be measured in the order of millimeters. All right, so the fundamental change that the Pixie assays are bringing to immunoassays is that we use a nanoporous substrate that minimizes the interaction distances between partner molecules. in this cartoon, we're showing kind of a cutout, an interior of a glass microchannel. And that microchannel is filled with a polymer substrate. So that polymer substrate acts as a nanoporous sieving matrix, and we're able to then functionalize this polymer backbone with capture molecules that are suspended at homogeneous intervals in 3D space. So think of it, the material is a gel. It's similar to gels that people are familiar with from slab gels, Western blotting, et cetera, except it's contained at a microscale within kind of like a capillary structure or a microchannel. By decorating the polymer, this kind of polymer mesh with the capture molecules, we're able to minimize the interaction distances that are seen between two partner molecules, between antibody and antigen, for example. Okay, so we have this functionalized nanoporous capture matrix. Secondly, Correlia combines this unique capture substrate with principles of electrophoresis. So by combining the capture substrate with an electrophoretic driving force, we're able to accelerate the probability that two partner molecules are going to interact and bind. I think most life scientists are familiar with the basic theory behind electrophoresis, which is that under the influence of an electric field, a charged molecule will migrate in the direction of the opposite charge. So within the Pixie cartridges, we use this electromotive force not as a method of protein separation, but instead it's a way to drive the target molecules through the capture gels. So we'll just show a cartoon first, and then next slide we'll see the real thing. By creating an electric potential across this gel-filled capillary, we're able to drive the antigens to get stuck to the antibody molecules on a really unprecedented time scale. So the characteristic length scale, the interaction distance between the antigen and the antibody, goes to nanometers instead of millimeters. And so let's take a video, let's look at a video of how this works. So this video is slightly sped up and we'll play it a few times. From right to left, we see a fluorescent target protein that is being driven across the gel substrate. So from right to left, this is a process that occurs in about 90 seconds. You can see, we used a fluorescent target molecule, a target protein in this case, to illustrate what's happening within the gel. But you can see a high degree of binding on the right side of the gel, which illustrates the strong affinity of the antigen for the antibody. This protein in particular, I think this was a fluorescently labeled IL-1 beta cytokine. The technique is based around a fluorescence measurement. By measuring the accumulation of fluorescence signal within the capillary over time, we're able to then take a proportionate reading, and then the level of fluorescence detection is proportional to the target concentration of the molecule of the analyte that you're interested in. So the analogy that we like to make about the Pixie technology versus the ELISA is the ELISA is like fishing with a pole, whereas the Pixie is like fishing with a net. Now, both of them will work to get your fish, but one of these methods is going to be a lot more efficient. And not only do we have this three-dimensional capture substrate within the Pixie cartridge, but we're using the electromotive force of electrophoresis to effectively drive your targets into the net. By coupling this unique nanoporous substrate with the principles of electrophoretic transport, Pixie assays are able to overcome the limits of traditional ELISA and make advancements in efficiency on multiple levels. The Pixie cartridges are able to take microscale volumes from as little as 2 microliters of a diluted sample. The assays can complete in as little as 60 minutes, depending on the complexity of the assay. And we've built an entire system around the cartridge that allows assays to be automated in a sample in, answer out format. So we'll take a look at kind of the basics of how the platform works later. I also wanted to show how the Pixie assay also generates measurements with expanded dynamic range. I think this is an important point. And the expanded dynamic range is partially due to the enhanced binding capacity of the substrate. This gel is able to, have a more dense capture of molecules versus what a microliter plate can do. But it's also partially due to the Pixie system's unique fluorescence detection and image processing techniques on the back end. So this example shown here shows a typical, cancer research protein. This is a IL-1 beta again, and we're looking at the performance of the Pixie kit versus an ELISA kit from Abcam. You can see that the Pixie offers, sub-picogram per milliliters levels of sensitivity with an expanded dynamic range of 5 logs from a single dilution factor. Okay, so we're able to exceed the detection range and the sensitivity of many conventional ELISA tests. An expanded dynamic range is very useful for many users because scientists, if you have an unknown sample, you don't know how it's going to fall within the limits of detection for your particular kit, right? So scientists often have to run their samples estimating and applying multiple dilution factors in an attempt to hit the sweet spot of the linear dynamic range. Giving the users broader dynamic range means less guesswork on the part of scientists, less samples consumed, and less work involved to get the data that they really need. Okay, so what does the Pixie platform look like? The Pixie platform actually consists of a few major components. There's the microfluidic cartridge that we talked about. There is an electrophoresis module that sits within the deck of a liquid handling robot. There's a separate imager that's required to get the fluorescence reading. And then Correlia also has a comprehensive software package that coordinates each of those cartridge module imager components. And the software also does data analysis for you. So another really unique thing about the Pixie immunoassay platform and the module is that it is designed for compatibility in terms of the module and imager, like these two central parts of the diagram. The module and imager are designed for compatibility with a diverse range of robots, liquid handling robots and imagers. So some of the robots that we are compatible with are listed here. I think our first offering is compatible with the OpenTrons robot. And if you have any of the other robots listed here within your lab, Hamilton, TCANs, et cetera, please get in touch and we can help you to get the module into your systems. So this year, we are working with our customers to get these immunoassay modules into your robots, into your labs. The workflow of the Pixie system is as follows. So assuming that we have the liquid handler and the module set up, and there's only a couple steps that the user needs hands-on involvement for on the front end. First is the loading of the diluted samples, placing the labware into the deck of the robot. Secondly, the Correlia Pixie cartridge goes into the deck of the robot as well. After those things are accomplished, the assay is basically hands-off. Sample loading, all of the steps are carried out by the liquid handler, and with other steps programmed, from the electrophoresis unit. So at this point, after the samples and the cartridge are loaded into the robot, the user can take a break, can take a coffee break, or they can, they can also put down the input, kind of the layout of the labware, and that would allow the user in a later step to be able to process the biomarker data. So it really is a sample in, answer out workflow that can complete in as little as 60 minutes, depending on how many samples you're running and the specific kit that you're running that day. Okay, so let's take a look at this instrument in motion. So over the past couple of years, Correlia has been serving many of the top 10 pharma companies in the world. So this video is just going to show the system up and running within Correlia's labs. So everything is automated. Sample delivery is all carried out from the liquid handler side. Other functions that are automated include image processing and data analysis. So over the last few years, our company has been serving 5 of the top 10 global pharmas. And this year is a big year as Correlia transitions from being a service provider to an instrument provider. In terms of the kits that we're offering, I'm going to take a closer look at a couple of case studies in which we are doing IgG measurements of therapeutic antibodies from small animal models. Small animal models are an especially interesting case for Pixie customers because the reductions in sample volume really allow for our customers to get more granular data from small animals. So the case study that we're going to focus on is a pharmacokinetic study of therapeutic, monoclonal IgG1 or IgG2 in a small animal model. So taking a PK profile of what this drug looks like in the bloodstream over time. With traditional immunoassays that require up to 100 microliters, the small size of the mouse is prohibitive, and it actually inhibits a lot of the data that scientists want. So for a time course, it is not uncommon for scientists to use a different animal specimen that needs to be sacrificed to represent individual time points. So this is not only cost prohibitive, but it also presents more variability. As we know, there's inherent variability from animal specimen to animal specimen. The microscale capabilities of the Pixie system allow for longitudinal sampling. If you can just microsample from the same animal, then you can follow that animal over a time course and not only reduce your costs, but have a more realistic depiction of the breakdown of that drug over time. We're going to be looking at a couple of case studies in particular. The first one that we're going to be looking at is a generic IgG1 titer measurement. So I want to take some time here to kind of illustrate the sandwich immunoassay concept. So even though there are some major advantages and increases in efficiency in what is going on within the Pixie cartridge, the underlying sandwich format remains unchanged. So, it's a traditional sandwich, capture antibody, target, detection antibody, right? And we can functionalize the kit to any user's needs. And the capture molecule is actually linked to this proprietary nanoporous substrate through a biotin link to the polymer. Okay. So we can design these kits with specific customer applications in mind. The first one that we'll be showing is a generic anti-IgG capture molecule. And then the second case study that we'll be looking at is a more idiotype-specific antibody measurement where there's a bait ligand that's immobilized to the polymer. We're going to look for a specific IgG target based on affinity to that ligand, and then use a fluorescence detection antibody in both cases. So we'll be looking at both generic and ligand-specific forms of antibody measurements. Again, it's like a standard sandwich concept, but in a kind of improved footprint, and improved format. So looking more closely at the case study, the first case study of a generic IgG1 measurement, the target that is examined in this case is IgG1 therapeutic from rat plasma. And so in this case, you can think of an animal model that is being dosed by a human monoclonal drug in a pharmacokinetic study. The specific performance characteristics that we're going to examine in this case include dynamic range, repeatability, recovery, and we're going to correlate the results against a traditional ELISA. Okay, so to demonstrate the dynamic range of this assay, we created standards that span across 5 orders of magnitude. These standards range from the single-digit nanogram per milliliter on the low end to up to 25 micrograms per milliliter on the high end. This represents kind of the relevant physiological range of antibody dosing in a mammal, therapeutic antibody dosing in a mammal. Okay, so we're able to achieve this dynamic range. We're also looking at, okay, from day to day, how much does that standard curve vary over time? To characterize that repeatability, we looked at the day-to-day variation of the standards, 3 different runs across 3 different days. The average day-to-day CVs across 5 orders of magnitude was 11% in this case. And then we also looked at the recovery, and we defined that as, you know, the measured concentration of the antibody over the known concentration of the antibody. So again, we're looking at samples. These are spiked samples of known concentration across 5 orders of magnitude. We see that the average recovery from Correlia's Pixie system was 87%. So that's, you know, within 15% of the known value, if we take the spike concentration as the known value. Average CVs for these measurements was 8%. Okay. Next, we want to kind of check out how the Pixie immunoassay results stack up against the ELISA. So first, we're going to look at Pixie results relative to the known concentration of these mock samples. Then we'll take a look at the same analysis, but for an ELISA kit. And then finally, we'll go head-to-head, compare the Pixie against the ELISA. With the Pixie system, we see that across these 5 orders of magnitude, we're getting a really, really nice linear dynamic range. So, you know, immunoassays, they characteristically show this kind of S-shaped curve, right? But in this range illustrated here, we're totally linear. We don't see that flattening out on the low end or the high end yet, with a really nice R-squared value, 0.999. ELISA, on the other hand, is okay, it's a little less linear. I think one of the challenges of working with the ELISA kit is that it does have a more narrow dynamic range, right? So in order to accommodate the more narrow dynamic range of the ELISA kit, the user has to select multiple dilution factors. By applying multiple dilution factors, the user can then kind of normalize the results to the starting concentrations. So if we're looking at this set of ELISA results, you might notice some nonlinearity there. And so that nonlinearity it can be attributed to the 2 different dilution factors that have to be applied for this set of samples to work with the ELISA kit. That nonlinearity also contributes to the lower R-squared value. So, you know, the narrow dynamic range of the ELISA necessitates the use of 2 different dilution factors. And that's a common problem that we see with ELISAs. So R-squared value, about 0.95 there. And when we look at the comparison of the ELISA versus the Pixie immunoassay system results, we're also seeing an R-squared of about 0.95, 0.947. That nonlinearity at the high end is still present, and that can be attributed to what we saw previously when we looked at the ELISA versus the theoretical concentrations. But overall, there's good agreement between the ELISA and the Pixie results. So we're demonstrating, you know, this IgG1 assay from rat plasma, broad dynamic range, almost 5 orders of magnitude, limit of detection that's, you know, single-digit nanogram per milliliter. That's kind of typical for this type of, you know, therapeutic IgG application. Intra-assay CVs less than 10%, average recoveries within 15% of the known values, and a strong correlation against traditional ELISA format. Additionally, I want to just highlight some of the practical aspects of how this assay was run in the Correlia system. We're able to get, you know, almost 5 logs dynamic range from a single dilution factor. The assay only required 2.5 microliters of the diluted sample per data point. And if we were applying a 10x dilution factor, in this case, that means that only 24 microliters of a raw sample could be applied to generate 96 unique protein measurements. Now, I talked earlier about how the kits can also be customized to provide more subclass or ligand specificity. So I just wanted to show one more data set in which we're able to kind of customize the assay by swapping in different capture antibodies. So in the next data set, we're going to take a look at results when an anti-IgG1 antibody is applied, is kind of linked to the polymer, versus results in a different kit where an anti-IgG2 is linked to the polymer. So one application case for this kind of subclass-based specificity could be if an investigator is looking at an animal model with a cocktail of 2 or more therapeutic antibodies of different subclass and seeking to differentiate the profiles of these, 2 monoclonals over time. So in this example, first we'll show the kit that has been customized for IgG1 specificity. So when that kit is applied and we look at IgG1 versus IgG2, you see a high signal to noise ratio for the IgG1 target and very low cross-reactivity to the IgG2 sample, and both targets are measured at high concentration, 20 micrograms per milliliter. Similarly, we can do the reverse examination where we're using an anti-IgG2 capture and testing cross-reactivity. We see high signal to noise for the IgG2 target and a very low cross-reactivity when that capture antibody is run together with an IgG1 target. Okay, so let's take this one step further then. Instead of just using, a generic anti-IgG capture molecule, let's take it one step further and look at a case study where we're doing a more idiotype specific measurement using a ligand that is known to bind to an anti-PD-1, for example. So this is an experiment that we ran together with a third-party vendor. And they not only were able to supply us with components of this sandwich, but they also ran a comparison experiment for us on the MSD Immunoassay platform. So again, specifically, this is an anti-PD-1 therapeutic antibody target from mouse serum. It was spiked into mouse serum, and then we used those spike samples to characterize dynamic range, repeatability, and correlation against the MSD Immunoassay. First, looking at Correlia's Pixie measurements of the anti-PD-1, we're able to get a 4-log dynamic range, lower limit of its detection below 1 nanogram per milliliter, good R-squared value, and a CV of 7%. When our collaborators took the exact same antibody pair and moved them onto the MSD platform, we're seeing comparable dynamic range, limit of the detection that's a little higher, CVs that's a little higher. So this example demonstrates how if we have reagents that are working well in plate-based formats, it can be directly translated into Correlia's electrophoretic immunoassay and achieve equivalent or better results. In such, with such customizability, kits can be assembled from basically any working sandwich pair. And then the other advantages of the Pixie system are then brought to bear, right? In specific applications, the Pixie system can expand the possibilities for biomarker and PK/PD measurements. There's this microvolume sample compatibility, which opens up different kinds of precious samples for further analysis. There's broad dynamic range. In some cases, we've achieved over 6 logs of dynamic range at a single dilution factor. A fast automated workflow that's made possible by the integration with a liquid handling robot. High precision measurements that we generally characterize through low coefficients of variation. So the data set shown here is a collaboration with a major, a top 10 pharma company that provided us with therapeutic IgG1 samples. In this case, running the IgG standards within Correlia's system provided CVs of less than 5%, 3% in this case. So we have been, really happy about the opportunities that we've had to service major pharma companies, as shown here. Over the last couple of years, Correlia has actually served 5 of the top 10 global pharmas, as well as major global CROs. I think these customers have really seen the need for proteomic tools to produce more data in a more reliable way and shifting it towards automation. Some of the innovations that are accelerating a need for proteomic data include precision medicine, where increasingly we see that patient samples are screened against a number of drugs to find the best individual treatment options. There's gene therapy applications where protein biomarker tests are used for quality control to determine the efficacy of gene delivery. There's also cancer drugs where many of the most promising therapeutics, the billion-dollar drugs, are biologics. And of course, in these days of the pandemic, I think everyone, including everyone's grandmother, knows about antigen tests, antibodies tests. These really highlight the importance of protein science in public health. So, today I was able to discuss one set of applications around the small animal models. There's really a lot more that I wasn't able to share this morning. For more papers and application notes, I encourage you to visit our website, correliabio.com. I'll just highlight one white paper in particular, the one in the forefront here. You'll be able to get it off of our website, I think in February. I don't think it's online yet, but in February 2022, you can look for more technical materials that accompanies our product introduction. The white paper in particular that's shown here is an interesting case study for those who are interested in cytokine measurements. In this case, there are IL-1 beta measurements from volume-limited renal carcinoma samples. So there are other, applications outside of small animal models, such as microbiopsies, cell culture, where the ability to work with small volumes really opens up more types of measurements and more protein data. In the pipeline here, upcoming, I think we're going to be focusing on two classes of molecules for our product launch this year. That's cytokines and therapeutic antibodies. I was only able to talk about therapeutic antibodies today. And that's basically it. I think we're excited to demonstrate some of these results for you today. The expanded capabilities for biomarker PK and PD measurements, the microsample compatibility that's made possible by the accelerating of protein interactions within the Pixie gel and cartridge, broad dynamic range, fast automated workflow. And importantly, we are going to be doing an introduction of this instrument very soon in February at the SLAS conference in Boston. We actually won an award at SLAS 2020 pre-pandemic for the best startup that year. And this year we are awarded we have been named as a finalist for the new product award in 2022. I didn't get to show a lot of the details about the software and the instrument, but I think all of that will be forthcoming soon. There are opportunities for early access and collaboration, especially if you already have one of the robots that was listed on one of my earlier slides. And if you have a common liquid handling robot in your lab, we'd love to talk to you about how we can place the module into your system and get these immunoassays up and running for you and accelerate your organization's proteomic data collection. My email address is here, and I'm happy to take questions at this time.
Thank you, Samuel, for that excellent presentation. We certainly appreciate that. And since you finished with talking about the handling instrumentation that's compatible with your cartridges and your technology, I want to jump to a question real quick from a person by the name of Courtney Alberry. So Courtney says, for the module compatibility with Hamilton, do you have to have a dedicated Hamilton for the module that is installed in it with regards to the deck space, or can it be removed temporarily to run other liquid handling scripts and methods on the Hamilton?
Great question. Yes. The module is designed to be removable. There are no permanent fixtures that are required to install the module on your robot deck. So it is removable. That said, there will be some calibration that's required every time you remove the module and replace the module. So maybe if you want to save time, I think a lot of our customers would want to leave the robots on their deck, or sorry, the module on their deck. But I understand that every lab has different needs, and if your lab, has a shared robot, then it is possible to remove it and open up that space for other people who are using that instrument.
Great. Thanks for that answer. And I want to get to some of the more interesting questions. It's a fascinating system that you have there, because you combine a nanoporous gel substrate with electrophoretic driving forces. And Jerry Ping had what I thought was a very good technical question. He asked, could your system be biased towards certain types of proteins regarding their electrophoretic properties over other types of proteins?
Mm-hmm. Yes, that's a great question. And I think that reflects the audience members' understanding of protein science. So it's true, different proteins have different isoelectric points. We use a native form of protein electrophoresis that does not denature the proteins. Correlia's kits are designed for specific targets. So for example, just like you buy an ELISA kit for a specific target, Correlia's kits are designed with one unique target in mind right now. And that means that we are also able to design the buffer system to ensure the electrophoretic mobility of that protein. So the audience member is totally right. Different proteins have different electrophoretic mobilities. The ability to tune the buffer for the specific protein of interest also kind of can serve as a selective mechanism to make sure that we mobilize the proteins of interest and we minimize, you can't eliminate it, but you minimize the interference or the mobility of other proteins that might be present in your sample.
Great, great. You mentioned your kits are primarily designed against specific targets. I have a couple of questions here. One is from Bradley, and he asks, what's your throughput regarding samples per day? And then another related question is it multiplex capable?
Yes. Okay. Maybe I'll start with the second question first. It is multiplex capable. But for our first products, we are focusing on one at a time. I think in our portfolio in the future, we will be thinking about which ones we want to put together into multiplex panels. And I think that will largely be driven by customer feedback and application areas, the specific application spaces. But you can imagine, for example, maybe we have a cancer biology panel, 4 cytokines that are of high interest there, or a inflammation panel, right? The other question I think had to do with the time scale, right? So I think I intentionally did not put a lot of information about the time scale. It is true that we can get assays completed in 60 minutes. I wanted to mention that most of that time, of that 60 minutes, is actually liquid handling time. It's your robot and so the answer will depend what kind of robot you have, how many samples you're doing. But if we think about, the steps that are required to run an immunoassay in terms of incubation steps, and anything that required an incubation step that, took hours, anything that required hours of incubation on the, in a traditional format, those incubation steps now occur on the order of seconds, really, on Correlia's format. So if you take the liquid handling steps out of the equation, the assay completes in well under an hour. But with a typical, liquid handling robot, I can give an example of, the system that we have in the lab right now. Typical liquid handling robot that we have in the lab right now runs 96 samples in about 2.5 hours, I'd say. Yeah, but your results may vary depending on what robot you have.
Right. It's really amazing that you can literally get the sample volume down 50-fold lower than what would be typical for an ELISA. I noticed that in your mouse animal model experiment. Jill Ulick asked, what is the dilution factor that you use on your samples?
Yeah, great question. Typically 10 to 20x. Yeah. But of course there is some flexibility there. I think in some minimum required dilution experiments, we have been able to get it to work and as little as 2x diluted samples, a 1-to-1 dilution of sample matrix and buffer. But of course it will be sample matrix dependent. But typically I would say for serum, plasma, cell culture, 10 to 20 is most common.
Great. And regarding the types of proteins that work better than others, one of the first questions we got was from Rodrigo Yellen, if I pronounced your last name right, Rodrigo, is, does it work well with membrane proteins?
Great question. Yeah. So, I don't think it is optimized for whole cells right now. Of course, for membrane proteins, it really is going to depend on the way that you're solubilizing that membrane. So I'd say working with membrane proteins is going to be trickier, and that's more of an active area of R&D for us right now. Not that it can't be done, but I think the solubilization method is going to be really important there.
I have a technical question because I'm fascinated with the nanogel substrate. Is it the physical properties of the gel, or is there some special chemical property of your gel that causes a reduction in the distance between the analyte and the antibody? Or can you just tell us a little more without breaching trade secrets how that works?
Sure. Yes. So I'll start with the example of a traditional slab gel, like a polyacrylamide gel, which I think most biologists are familiar with. Those are tuned such that so that you can separate proteins based on size, right? And so I think a typical slab gel might have a polyacrylamide percentage of around 6%, 4 to 8% is kind of optimum for sizing. Because Correlia is not seeking size information with our Pixie gels, we're able to go to a lower gel density. And the gel itself, it's basically a modified form of polyacrylamide that has streptavidin sites on it, right? So we are able to drop the density of the gel so that it doesn't inhibit protein migration. We actually want proteins to flow through the gel as quickly as possible. So in terms of optimizing the gel density, we are able to kind of tune it for most efficient protein transport. And then secondly, the other knob that we can turn to specify the density of the capture molecule is basically how many streptavidin attachment sites are present within that gel. Yeah, so I think this nanoporous matrix, it's actually pretty simple. And the proprietary coverage that Correlia has on this deals with the method of functionalization. Yeah, but we basically, we've done experiments to optimize the density of the capture molecule, which is suspended in the 3-dimensional gel matrix.
Very powerful combination of electrophoretic driving force and nanotechnology. Chandra Raman asked, does the system also work with small molecules or just biochemical-based proteins?
Yeah, it can work with small molecules as well. So for example, we could do nucleic acid affinity enrichment. That's one possibility. But in our first product offerings, Correlia is very focused on the proteomics market because of that lag that we see between the genomic side and the proteomic side. I think there's I think I probably don't need to tell the audience of this seminar series that there's really untapped potential there. And that's why we focus on protein measurements first. If there are other, small molecule applications that you're interested in, Correlia is definitely open to discussing those possibilities as well. So we would welcome those conversations.
Great. Lisa Woody has a good question here. Are there any general limitations on how samples can be prepared, like the detergents that you use or denaturants and things like that?
Yeah, that's a good question. I think as a rule, our immunoassay kits have been designed for native form proteins. So that kind of makes me think of the earlier question about membrane proteins. I know, a lot of the methods for getting proteins out of membranes involve detergents. So those can be trickier to handle. I'd say the types of samples that we have worked with to date include cell lysates, cell cultures, and tissue samples such as plasma and serum. Yeah, and so in our first applications, we've focused on native form proteins. It's not that we can't work with them, but some of the things that we look out for in terms of the sample is detergents and salts. High salt content often needs to be diluted appropriately in order to ensure compatibility.
Great. I have a question from Hal Tung, who I think I know, so I definitely have to ask this one. Is typically how much antibody is needed compared to ELISA?
Oh yeah, that's a great question. It's orders of magnitude less. So like if I don't have a bank scale here, I've neglected to put in a scale bar in this cartoon, but the volumes of antibody that we are using to functionalize the cartridge, if you look at the area that's actually functionalized, it's more like nanoliter scale, right? Not even microliter scale. It's more like nanoliter scale. And so there are also significant savings that can be achieved on the reagent front as well. So yeah, it's an orders of magnitude less.
Great, big cost savings there.
Mm-hmm.
And I want to get to this question. It's a little bit long, but I'm going to try my best here. It's from Kavita Sharma. And Kavita asks, how would the IgG subclass be detected via Pixie, being more sensitive than chip assay in terms of setting the different primed population of IgG, specifically in case of a viral invasion? I know that's a tough one, but maybe you could do your best.
Yeah. So I think by chip assay, I assume that means
I think she's meaning chromosomal immunopresentation, which is incredibly sensitive. What we can do is we can have Kavita ask you that offline if you like, because that's quite an in-depth question.
Yes, yeah, I'd like to get into that with her. Maybe separately, we can dig into the nuances there.
Sure, no problem at all. One last question for you, because we're just out of time. I think we got to just about all the questions. If we didn't get to your question, we can forward it on to Dr. Kia. And that question is, how has cynobiological products worked in your system?
Great, yeah. I would say one of the areas where we are most interested in partnering with Cyno right now is to bring Cyno's matched pair antibodies onto our system. So, Correlia does not generate our own antibodies, right? And so actually a significant portion of my time on the scientific development team is screening antibodies, and there's a lot of not good antibodies out there. So it means a lot to Correlia if we can find trusted antibody partners with well-validated pairs. Those, I think, will find the most success in our customer hands, and also they will streamline Correlia's internal assay development process. Yeah, so we were happy, when we talked to Sino, to find that they have a portfolio of known working match pairs available to us.
Great, thank you for that, Samuel. We appreciate it. There, all of our antibody pairs and all of our products are manufactured 100% in-house. We don't relabel any products, and I just want the whole audience to know that. And we appreciate your kudos on the quality and reproducibility of our antibody pairs. So with that, I think I'll wrap everything up. First, I'd like to thank all the attendees. We had a fantastic turnout today. We appreciate your time, no matter where you might be in the world. Thank you for attending. Certainly, thank you, Dr. Tia. It was a fantastic presentation. Congrats to you on your success with Correlia. I know you're a founder there, have been there a number of years, and have really taken the company to another level. And so it's just wonderful to hear about your technology and hear how Sino is somewhat impacting what you do at Correlia. And I also want to thank my colleague, Christine Lay. Christine is a marketing specialist at Sino Biological, and she's the one who set up and really executed this webinar. And with that, again, thanks to everybody who's on right now and attended. And we will end this webinar and look for the next one on the Sino Biological website. You can register there today. We'll have another one in just a few weeks. So with that, thanks again, Samuel, and we'll see you. Zai Chan.
Yeah, thanks, Rob. Appreciate it.